This is your lecture on tissue culture and stem cells. Tissue culture is the growth of animal cells in culture. It can also apply to plant tissue, but most often you will see it used in terms of animal cells growing in culture. To be able to keep those animal cells alive in culture, you need to be able to replicate the environment as closely as possible that they would get in a living organism. So to that end, when you have these individual cells growing in culture, there are 9 amino acids that cannot be synthesized by vertebrates, for example, and 3 that are difficult to synthesize. So you need to have a rich media for them that provides all the amino acids that they're going to need to grow. In addition, any vitamins and minerals that they're going to need to grow. Rich media also contains glucose as a carbon source. So in the same way that our cells use glucose to fuel our bodies, tissue cultures will also use the same things to keep their cells running. Another thing that our blood supplies to our cells are specific types of growth factors that keep us alive, and it's produced by our different organs and put into our blood supply. Well, if you have an animal cell growing in culture, it's generally going to be from a single organ or a single type of tissue from the body, like skin cells or something like that. And so as part of this rich media, you need to add in those growth factors in something called the serum. So most frequently, it's something like fetal bovine serum, and fetal bovine serum comes from very, very young cows, like just born cows, and from our veal industry here in the United States, and they are bled, and the serum is separated from the red blood cells. And the serum, because they are a young growing animal, the serum is going to contain growth factors. Now, it's also important to note that depending on the kind of cell you are growing, that fetal bovine serum may not be the only serum that would be appropriate for your cells. We used to grow chicken cells in Dr. Stump's lab, for example, and fetal bovine serum did not work, but we had to get special chicken serum. So sometimes cells may have different specific needs because of the different growth factors they need to survive. It's also important to know that different amounts of serum might be needed. So you might have to have 10% fetal bovine serum as part of your media, or 5%. So that can also differ. And usually when you buy a type of tissue culture cells from a company, they will tell you what kind of media is needed to keep your cells growing, and they'll also be happy to sell you the fetal bovine serum that contains all the growth factors that you need. But there's other companies also that will specialize in, here's just our fetal bovine serum. And sometimes different growth factors are present in different lots of the serum. So, for example, in Dr. Stump's lab, when we were not messing around with chicken cells, we were growing Drosophila embryonic tissue culture cells, and those also had specific growth factors that they needed. Now, fetal bovine serum worked fine for them, but there was different lots of the fetal bovine serum from different companies. Some lots would work, and some lots would not. And we were never really able to nail down what made one serum work well and another serum not work well. So when we needed more fetal bovine serum, we would test out, say, 6 different lots from 3 different companies. We'd find one that worked well, and we'd order 100 bottles of it just to keep us in business for a while. And then 2 years later, we'd have to do, the same thing all over again, because it was such a huge undertaking. If you're lucky enough to be having a type of tissue culture cell that grows well in any fetal bovine serum, bonus. So, cells are growing on special solid surfaces. Now, a normal cell that grows in your body has what's called contact inhibition. When it comes in contact with another cell, it stops growing. But the type of cells you're going to find growing in tissue culture cells if it is an immortal cell line at least, it has lost generally its contact inhibition, but it still needs a surface on which to grow. So there are special types of plates that have been treated with collagen or negatively charged groups, and they help other cells adhere to those plates. So if you've ever seen, for example, a petri dish, if you took like a biology lab, maybe bacteriology, and you were trying to use a petri dish to grow bacteria, even though the tissue culture plates look similar, the petri dishes that you would do in bacteriology would not work for tissue culture cell because they have not been treated properly. So again, companies are more than happy to sell you special surfaces that have been treated properly to help your tissue culture cells grow, and they're a lot more expensive than the normal petri dishes in terms of price. So a case of regular petri dishes for bacteria might be $35 a case. A case of plates for tissue culture cells, 200 to 250, and that's our discounted price here at San Diego State. So I'm sure other people get charged a lot more for them. Now, if you're talking about a primary cell would be, we could take a cell from your body and we could actually grow a cell from your body on a tissue culture plate. We would add trypsin to separate the connections between the cells, and we could put it onto tissue culture dishes with that same rich media, and cells from your body would grow and divide about 50 times. And then after 50 times, they would start to die off. So the difference between primary cells and an immortal cell line in culture is it's been transformed. And to allow them to grow indefinitely, there's been some kind of an oncogenic genetic change, very often seen, most often, in cancer cells. And this has a change to the cells somehow, which allows them to divide far more than 50 times. We saw this in HeLa cells. HeLa cells were the first regular growing type of tissue culture cells that were able to grow indefinitely in culture. There's a fantastic book called The Immortal Life of Henrietta Lacks, all about the HeLa cells, which came from Henrietta Lacks. The book talks about the ethics of having taken cells from her body to grow them in the interests of research. It talks about the growth of tissue culture and everything that it's been used for. And it's just a fantastic book, and I highly, highly recommend reading it. So when we're talking about different types of cells, one thing that people noticed is that sometimes embryonic cells gave rise to specific types of cell lines. You can get cells at an early enough phase, and they seem to have the ability to grow indefinitely. They almost seem like cancer cells, even though there was no direct oncogenic change that could be attributed to them. So just some pictures here. This is a media bottle. So this is a bottle that's been specially treated on the inside to allow cells to adhere to it. And this is a picture of the rich media inside, and cells grow along that bottom surface. And then there's a little filter in the cap, and this cap allows the exchange of gases, and I'll be talking about the gases in a moment that are required for tissue culture cells. Here is what tissue culture cells sort of look like when they're growing on a plate. They're individual cells that sort of stretch out so that they have plenty of space around them on a plate. Eventually, as they keep growing and dividing, they become a little too crowded. And if they become too crowded on the plate, you will run out of the nutrients in the solution, and then all of your cells will start to die. So it's important to keep dividing your cells on a regular basis if you are trying to grow them to have them survive. So some of the considerations for working with tissue culture cells is, are you going to grow them as adherent cells? And that was that picture of cells that were sort of spread out on the bottom of the plate. But some type of cells also will grow just fine in suspension. So they'll grow just as little round cells without ever adhering to a plate at all. So, and they might be very loosely adherent. So as an example, our Drosophila embryonic stem cells can be trained to grow either as adherent, or you can grow cells in suspension, because even when they're adherent, they're so very loosely adherent that even just shaking the plate will enable them to be in suspension in this rich media. As you get a greater number of cells, it's important to split them, and this is called passaging. And if you don't passage your cells, as I just said, they will run out of nutrients in the media. So you have to usually use trypsin to again take them off the surface of the plate to break all those connections, and then you can divide up your plates, your cells between multiple plates, and you will put new media on there. Now, some other considerations besides making sure that they have enough nutrients in the media: cells need to be growing at the proper temperature. And it depends on what kind of a source they're from. HeLa cells like to grow at 37 degrees because that's pretty much body temperature. But if you're growing tissue culture cells from Drosophila are insects, and those are used to, because they're not a warm-blooded creature, they're used to growing at basically room temperature. So you can grow Drosophila cells at 25 degrees, for example, but if you were to put Drosophila cells at 37 degrees that works for HeLa cells, they're all going to completely die. So you have to know what temperature your cell will grow in. You also have to know the best pH for your cells, and there can be some differences in pH. And that's why when you are purchasing cells or getting them from someone, they will usually tell you the best conditions to grow your cells in, what temperature to grow it at, what type of media to use, which has a certain pH, and things like that. Cells need a particular level of humidity in them, and I have a picture of an incubator, and you'll see in just a minute. And so you need the cells are used to being surrounded basically in an aqueous environment in the body, so you need humidity around the cells. If they get too dry, they don't have enough humidity, they're not going to grow well. Also, when cells are in your body, there's a much higher carbon dioxide level in your body than there is in the outside atmosphere. So generally, for mammalian cells, as an example, it's necessary to add carbon dioxide in the environment to support the levels of carbon dioxide that the cells need to grow. If you don't add carbon dioxide in, your cells will not thrive and may not even survive if the CO2 level gets low enough. Generally, you grow them in a dark environment because light can sometimes set up toxic reactions in the cell. So all of these are some considerations that you have when growing tissue culture cells. There's specific equipment for growing tissue culture cells. One of them is what's called a laminar flow hood. Now, you've seen hoods, in the chemical laboratories, but this is a different type of hood. It's not a hood which makes sure to suck air in from the outside, in through a hood up here. It instead puts a cushion of air down the front of the hood to make sure that the outside air stays out, so that the inside environment can remain sterile. So it functions completely differently. So you can't just use any old hood to grow tissue culture cells. It has to specifically be this type of laminar flow hood designed to keep the inside environment sterile. You also have a special carbon dioxide incubator. So I talked about how you need to put carbon dioxide in. So there'll usually be like a carbon dioxide tank to the side of it that's pumping CO2 to a different specific level, and you can check on your controls to make sure the CO2 is giving the right amount. And usually there's a tray of water at the bottom to give the proper humidity level for all the cells. And then you can see the growing bottles or growing petri dish type plates, but special ones for tissue culture cells are in the various shelves of the incubator, and you can have different types of cells growing in different incubators, or if they're compatible, different types of cells growing even in the same incubator. You also need to worry about how you're going to preserve your cells. To be able to preserve your cells long term, you need to be able to freeze them down. And to be able to freeze them down, you're going to freeze them in liquid nitrogen. But you can't freeze them too fast, so you have to be able to freeze them without causing microfractures in the cell membrane. So you usually put them over some liquid nitrogen vapor to allow them to come to a low temperature very slowly, and then you can immerse them in liquid nitrogen so that they will be preserved, and that you can preserve them this way for 10 years or even more, so that you can make a cell line, get it all preserved, and then take it and defrost it when you need that particular type of cell again. So there are huge tissue culture cell banks that you can order from if you're looking for a particular type of cell. There's huge catalogs for all these different types of tissue culture cells that have ever been used, from what type of animal they are and the conditions they need and stuff like that. Now, some special types of tissue culture cells are stem cells. And stem cells basically have the potential to develop into many different types of cells. And because they can develop into many different types of cells, they have the potential to become an internal repair system, because they can develop into multiple different types of cells. This is called differentiation. So you can take a basic embryonic cell and it can differentiate into multiple different types of cells. So a blastocyst is a 3 to 5 day old embryonic cluster of cells, and the blastocyst, all these embryonic cells in the blastocyst, eventually differentiate to give rise to the entire body of an organism. Some of them differentiate to form the hematopoietic system, the lymph system, the blood system. Some of them differentiate to form the various organs, to form muscles, to form skin. So these early cells give rise to the entire body of the organism. When you're working with stem cells, they have some very specific needs. So they're grown in cultures much like tissue culture cells are, but they need a little bit of extra help. So generally what you do with stem cells is you have a layer of other types of embryonic cells, very often mouse embryonic cells, that are used sort of as a feeder layer. And these mouse embryonic cells are treated so that they don't actually divide. But what they're doing is they're not able to divide, but they're still able to put out nutrients into the media and to provide a sort of a sticky layer for the stem cells to grow on, because the stem cells are not going to be able to adhere directly to the plates. You need some extra help for them. And when you're working with stem cells, you always have to characterize them to see if they have been differentiated, because remember, that's the job of stem cells, is to differentiate into all these different types of tissues. So you're constantly taking little bits of your sample and you're characterizing them by what growth factors are they currently putting out, to see if they start to differentiate. And if they start to differentiate, you get rid of those cells that are differentiating, and you keep the colonies that have not yet differentiated. So when you're working with stem cells, it requires a lot of very specialized work. Not very many people have the training to work with stem cells. Usually you start off doing regular tissue culture cells, and then you get special training to be able to work with stem cells. One of the ways that they see if they're differentiated, as cells begin to differentiate, they will put out different growth factors. So to see if they've differentiated, they take some of the cells and they break them open, and they would run them on an SDS page gel, just like you did in the aldolase experiment. And then they would transfer them to a membrane and do a blot on them. So maybe they'll have the same protein mix from the cells loaded in, let's say, 10 different lanes on a gel. And then they cut apart that membrane. And so all of those 10 different lanes, they'll do, one type of antibody on one lane and a different type of antibody in a different lane. And so what they're doing is they're saying, is this particular protein being expressed in the cells so that we can see if it has begun to differentiate. And so as they're starting to express different proteins, you can have antibodies against those different proteins, and you can detect the presence of them by doing a Western blot on all of these different colonies that you're trying to grow. So one of the very useful techniques that you've learned in this class is, if you are working with stem cells, you have to be able to do STS page and Western blots. Now, because the expression of the different factors are benchmarks for differentiation, it is also possible to do directed differentiation with stem cells. So lots of research is currently going on of using specific factors that are put into the media to force cells to become a certain type. So let's say, for example, you want to do an experiment with cardiac cells. Well, you've taken your human embryonic stem cells that you're growing, but you want to make sure that those embryonic stem cells don't just differentiate into any different kind of cell. You want them to specifically become cardiac cells. So you can use specific factors which are known to start different cascades in cells. You put that factor into the media, and that guides them. It does directed differentiation, so it guides them to becoming a certain type of cell, which you can then that you can then use in your work. So you will have colonies of stem cells that are undifferentiated, and then you're making sure to see if they're starting to differentiate, and then you can make them differentiate into specific types of things that you're interested in. Now, adult stem cells are also very useful. Current research is seeing whether they can have repair functions to them. So if you have an adult stem cell which is already differentiated, like say you already have a person, and this person has heart disease, and you want to be able to fix their heart. Well, to be able to fix their heart, let's say they've had a heart attack and some of the veins in their heart are no longer in good shape. You want to know whether, by putting in a certain type of cardiac stem cell, maybe from that person themselves, can we take stem cells out of a person? Can we make them into cardiac cells? Can we then inject those cardiac cells back into a person? And is this going to be able to help their heart repair? So this is the current status of research that's going on, taking differentiated cells and trying to say, can we induce them? Can we take these early embryonic stem cells and induce them? They have the potential to be pluripotent, which means they can differentiate into many different types of adult cells, and so they will induce them to become spinal cord type cells, or to become pancreatic cells, or to become brain cells for people who have Alzheimer's. So this is what they're trying to do. Can we repair spinal cord injuries by having cells that differentiate into nerve cells and injecting them into the spinal cord area? Sometimes that's worked a little bit with people, but there's no grand, aha moment. This always works for us. So lots of research is still ongoing. One thing that seems to have had greater effect is pancreatic stem cells. So there are currently people with artificial pancreases, not implanted inside of them, but sort of like in a little bag outside of their body. So the blood is directed to flow into this little bag that's outside of their body, which contains pancreatic stem cells, which are then producing insulin. So it's a way of sort of giving a person an artificial pancreas, and then you can occasionally replace this bag of cells on the outside of their body. as yet, we have not been able to recreate a completely working pancreas to be able to implant into someone, but things like this are a first step along that way. So generally, when working with stem cells, there are certain goals, and you want them to be able to proliferate extensively and generate sufficient tissue. We are there. We can make them proliferate. We can grow them in culture. We can generate lots of tissue for them, for people to work on. And you want to be able to differentiate them into the desired cell type. And we're also there. We can take embryonic cells and we can make them grow up into nerve cells or cardiac cells, pancreatic cells. And so what we want to be able to do then is have an entire organ that we can implant into somebody and that it will survive, that there are enough cells in it, that they're working well enough that it will survive in the recipient after transplant, and that it will integrate into the surrounding tissue and function appropriately for the duration of the recipient's life, with the overall goal being to avoid harm to the recipient because of the dangers of immune rejection. One of the things they're doing to try to fight immune rejection is trying to be able to uh, get some tissue from a person, and fat cells can harbor different types of stem cells, and so they're currently doing research on, okay, can we suck some fat out of somebody? I have a lifetime supply. Can we suck some fat out of somebody, and can we cause those cells through addition of specific factors to then differentiate into a certain type of tissue? And because we can do this from a person who needs that organ to be transplanted into them, it has all the immune factors that that same person does, so you can avoid harm to the recipient if those cells that are originally making the particular organ come from the patient themselves. Otherwise, if they come from somebody else, you have that immune rejection problem. So that's the status of stem cells, just a little bit of a primer for you. Here's what things that they have tried. So trying in an oral surgery, for example, when people are getting teeth fixed, as an example, trying to take some tissue and implanting it into a person who's getting an artificial tooth, for example, to help that tooth adhere into the jaw, or taking different types of tissue and growing them in a tissue culture and potentially causing them to differentiate into maybe someone who needs a new cornea, for example. And so if you can make a sheet of the cells, and then of epithelial cells, make an artificial cornea from someone, then you can try transplanting it, giving somebody an artificial cornea so that they will again be able to see. So this is the types of things they're trying. So far, we don't have anybody with 20/20 vision yet, but it has cleared up their vision somewhat with artificial corneas. They're still taking corneas from dead people, though, to really be able to give someone full eyesight. Other things, they're growing cells in sheets and not only trying to transplant them onto eyes, but ongoing research for heart and liver and kidneys. And what they're very successful at is growing skin cells. So currently, if you are a burn victim, for example, and you need tissue replaced in your epithelial layer so that you don't lose lots of fluids through your burned skin, they actually have artificial skin that is epithelial cells grown in tissue culture as sheets, and they will take these sheets and they will place them over the burned skin and and suture them down as a protective layer. And they don't have to worry about immune rejection because it's just the epithelial layer. So this allows your dermal layer of skin underneath the epithelial layer to be able to survive a little bit better and grow out. And then the artificial skin just naturally sloughs off as your own dermal layer starts producing more epithelial tissue. So they do that quite a bit now for burn victims. So there are companies who are in they manufacture these sheets of artificial skin cells, and they are actively selling them to hospitals. So that's another usage for tissue culture cells. That's it.